In a different lecture, we mentioned that all cancer is genetic, meaning all of it's due to changes in the DNA. And those changes in the DNA are called mutations, which you've probably heard of before. So we have to understand a little bit about how DNA works in order to understand why these mutations actually make an effect. So I'm going to show you, because DNA just holds information, but I'm going to show you how that information is important for an outcome in the cell, and then how a change in that information can change the outcome in the cell. So let's say this is some DNA. I'm just going to draw one strand of it, and I'm going to draw some DNA nucleotides. On this strand. Remember, each of these is just a different type of nucleotide, A, T, C, G. They're all attached together in a big long chain. And then there's a lot more in here that I'm not drawing because of time purposes. Okay, and the DNA keeps going on both ends here. All right, so this is part of this DNA sequence is what we call a gene, and a gene has a start and an end, okay? And there's a special start sequence of DNA that's the ATG sequence. You don't need to know that yet, but I'm just telling you that that's the start part. So here is the gene, because there's also end stop sequences. So we actually have a TAG stop sequence down here. So this is a gene, and this gene codes for a protein. So each grouping of DNA, it's in groups of threes that it's read by the machinery. Each group of 3 codes for a specific amino acid. So this one's called methionine, methionine amino acid. And remember, the amino acids are what's going to make up the protein. So they're the beads on a string for a protein, just like the beads on the string for the DNA is the nucleotides. A CCC code gives us what we call a proline amino acid. A GGC code gives us a glycine amino acid. So this is just when it reads the code, it knows what to build. And this is what ribosomes do, the part of the cell that builds proteins. So they read this code to build the protein. So isoleucine, ILE, is coded for by ATC, and CGG is coding for arginine. Then there'll be a lot more amino acids in here until we get to this TAG sequence, which is a stop sequence. It means don't put any more amino acids in. So this is just some amino acid here. Okay, so we have this long protein. And as we know, proteins fold up, and that's how they get their function. So let's say this protein folds like this. It has some spots that hold it together. Here's a spot that holds it together and folds. And then let's say here's a spot that holds it together and folds. Okay, so we have a certain shape of this protein. Now let's say, and that gives it a certain function. And that's because the folding. Okay. Now let's say this DNA is copied, because remember, before we divide a cell, we've got to copy the DNA in order to, have enough to make 2 cells. So this is the DNA copy for the new cell. So usually when we copy the DNA, it should be the same. So the machinery that does that, which we'll learn about in another unit, would just do the same sequence when it's copying it. But occasionally, there is a mistake made. So let's say here. So what I've shown here is that usually there's an A put in, and in this case, we've got a T put in. Now the cell is usually pretty good at catching these mistakes. So it actually proofreads its DNA after it copies it and makes sure there are no mistakes. But occasionally they get through. So let's say most of this DNA is fine. But we have this one change, and that, let's say that doesn't get caught by the proofreading machinery. So it accidentally gets through. It's like you accidentally turn your paper in with a spelling mistake. Sometimes this happens. The protein here still has methionine because it still has the same ATG code. CCC is the same for proline. GGC is the same for glycine. But now we have TTC, and that actually codes for an amino acid called phenylalanine instead of isoleucine. So you see there's a different amino acid here. It used to be isoleucine. So that's the mutation. The mutation is in the DNA. So this nucleotide here, this mistake, is the mutation. It's a DNA change. And the consequence of that mutation is that we've added a different amino acid. Now the next one is still arginine. Then we have a bunch more. There's an amino acid, and still we have our stop codon in the same spot. Okay. So what's the consequence of this? Well, let's say phenylalanine was important in that folding. So now we get this protein here, and when it folds, we'll go back and look at what it was before, it's going to be okay in the beginning. Then let's say this interaction doesn't happen anymore to hold it together. So we still have this interaction, but this other one, there's no interaction because the amino acid was changed. So let's say this is the phenylalanine, and it used to be here, the isoleucine. So now the phenylalanine doesn't interact the same way that the isoleucine did. And so what do we have? Potentially a different shape, and so potentially a different function. Now sometimes mutations don't actually change the shape, and sometimes even if the shape is changed, the function is still the same. But it could be a different function, or it could be no function due to this different shape, aka folding, different folding. So to summarize this idea, we have a DNA sequence that codes for a protein with a certain amino acid sequence. And the amino acids interact in a particular way. And that causes the folding of the protein. Into a specific shape. And that gives it some sort of specific function. FXN, that's just function. So that's what is originally going on. Then we have the DNA change, or the mutation. Remember, that's in the DNA, which can lead to something like a different amino acid sequence in our example. It could be one change like we showed. It could be many differences in the amino acid sequence. Even one change in DNA can actually lead to, if it's like an extra DNA letter, it can lead to a completely different code after that letter. So it can actually be a big change in sequence too, or just a single amino acid. So different amino acid sequence can lead to different amino acid interactions or lack of interactions, which means the protein folds differently or not at all. So we have a couple consequences here. One could be that the protein has a different function. One consequence could be that the protein has more than normal function, or it could have less than normal function or no function. And we could think about these types of mutation consequences in the context of cancer. More than normal function mutations are often what's leading to oncogenes. They're versions of the proteins that are too active. Less than or no function or loss of function is often what are mutated tumor suppressor genes. Loss of function. Let me add one thing here, is that it could be a different amino acid sequence. There's also another possibility, is that when you change the DNA, it codes for a stop too quickly. So it could be a different amino acid sequence. It can also be a too short amino acid sequence. So it stopped too early. And these can lead to, a too short sequence can lead to folding differently or not at all as well, and those kind of consequences. And often, though, mutations, changes don't lead to anything. Actually, that's most common. Most common is that mutations don't make a difference in the outcome of the protein. So they don't matter. But sometimes they do. And this happens naturally as part of the cell. Mutations are important because we need variation in the population. Like if you never had mutations, you couldn't make babies that look different from DNA changes. You'd never get any variation in the population. So mutations are essential for life, but we don't want them to happen in body cells, really. And we don't want them to happen too often, because that's when we can lead to problems. I'll also mention one more thing. Mutations don't have to happen in the protein region, protein coding region of a gene. They can also happen in what we're going to call regulatory regions. Those are regions that control genes, like turn them on and off, or how much the gene is on or off. So if you have changes in those regulatory regions, that can lead to genes being on too much or off too much, on at the wrong time, that kind of thing, which can also lead to these kinds of problems. So in that case, it's not actually changing the shape of a protein. In that case, it's changing how much protein is being made, whether it's too much or too little. So that's just a mention there that it can be in regulatory regions. So that's what a mutation is, and why it actually has consequences for the cell and what it can do and what it can't do and how it behaves.